So our center produces GWAS reports, and a GWAS report takes phenotypic and genotypic data, analyzes it in a standardized way, and then presents a series of figures to the user so that they can understand the results of their experiment. One of the cores of those reports is this. We call it a porcupine plot. It's a series of Manhattan plots that are superimposed on one another, identifying the regions where we saw statistically significant associations. You'll notice that when we see associations, we see not just one variant, one SNP associated, but a whole bunch of SNPs that are associated. And that's because nearby SNPs are co-inherited because they're in linkage disequilibrium with one another. And so that's the reason that you see these towers of SNPs. When we move to zoom in on a particular region, so now we're looking at just a small region of chromosome 7, this is called a locus zoom plot. And we're looking at the p-values for the association between SNPs, which are the dots, and a particular trait. You can see that some of the SNPs are extremely significant and they pass this horizontal line, the threshold for significance. You'll also see that the SNPs have different colors, and those colors are related to the p-values, but the color doesn't directly reflect the p-value. The color reflects the correlations between the SNPs, the linkage disequilibrium, which is a function of the SNPs being co-inherited with one another because they're physically close to one another. The red ones are very strongly correlated with the most associated SNP, and then yellow less so, green less so, and the colors of blue even less so. Because they're less correlated, those SNPs also are less significantly associated with the trait. In general, we'd want to look at the genes which are under the red SNPs, so in this case about 5 genes, which are under the most associated SNPs. That's probably the region that contains the causal variant. Thiago is now going to tell you a little bit more about these reports and how we get to the point that we can prepare one.
So after we receive the measurements of the animals from our phenotyping center, and we extract the DNA and extract the variants of this animal, we can try to associate the traits with the variants. And as an example, we can think does, in this case, this variant, would it increase the traits? And then we can make a measurement of just how significant is this effect. Once we have this effect, we can actually get a number as a p-value of the probability of the SNP being significant to influence the traits. And we're gonna do this for around 7 million variants. And we can make a plot like this, that we're gonna have variants per chromosome. And that's gonna be here for the p-value of the probability of mattering. Once we identify an area of interest, we can try to narrow down which is the most probable gene from a locus zoom plot. For example, in this plot here is from a study on attention. From this analysis, we might infer that one of the genes below this red area, where you can see the inflated p-value on chromosome 7, to have a significant effect on the attention. And once we identify the genes under this region in red, we can look for supported information, such as coding variants that might alter the protein itself, or expression variants that might alter the quantity of the protein within the cells. We can also look within the literature of those genes and look for associations in human GWASs or any pathway that is well described of these proteins that can be related to the phenotype that we are measuring. Given all the information that we aggregate, we can try to rank the genes and produce candidate genes and publish the set of candidate genes that collaborators can further validate.